Hi, everybody. For those of you that don't know me, I'm Carrie Bearden from UCLA, and so I can make this very quick because we don't really have results yet for the 22q11 working group, but I'll tell you about some preliminary stuff that I think is promising. And I want to start by just congratulating all the other working groups for the really incredible amount of progress everyone has made in a relatively short period of time. I think this is really exciting, so it's great to be a part of this. So just to orient you a little bit to what the 22q11.2 deletion syndrome is, if you're not familiar with it, it's also known as velocardiofacial or DiGeorge syndrome. It has an estimated incidence of 1 in 2,000 to 1 in 5,000 live births, so it's actually the second most common genetic syndrome following Down syndrome. So while it is a rare disorder, it's not as rare as you might think. So it results from hemizygous deletion on chromosome 22, and about 85% of patients have approximately the same 3 megabase deletion, encompassing about 60 known genes. And then there's a smaller percentage, about 15%, have a smaller deletion that's about 1.5 megabases in size. The reason there are some associated physical features, cardiac defects, craniofacial abnormalities are pretty common. The reason that I'm particularly interested in this disorder is it is the greatest known genetic risk factor for schizophrenia. So about 25 to 30% of patients with the disorder develop a psychotic disorder in adolescents or adults. There are also a high rate of other developmental neuropsychiatric disorders associated with it. So 14 to 50%, depending on the study, have autism spectrum disorder. Pretty high rates of ADHD and anxiety disorder as well in kids. So it's a really interesting disorder from a developmental perspective and also for looking at questions regarding neuropsychiatric pleiotropy. So these are just to give you a sense of what the volumetric findings look like. These are just data from our own single-site study in about 80 patients with deletion and 65 controls. And so these are effect sizes for volumetric reductions in patients compared to controls. And so you can see that comparing to idiopathic schizophrenia, the patients overall look a little bit different. So they have, and these are kids, average age is about 11 in the sample. So the inferior temporal gyrus in particular is reduced, and there's a lot of medial volumetric reduction and parietal occipital reduction. The frontal lobes are relatively preserved, which is interesting, and so these are cross-sectional data, but our longitudinal data actually show that there's a delayed developmental trajectory of frontal maturation, which is pretty interesting. And so the other thing from our single-site study that we found is that when we look at controlling for multiple comparisons, it was particularly a right medial orbitofrontal thickness that was predictive of positive psychotic symptoms in the deletion patients. And so it'll be interesting to see if this replicates across sites. So our 22q ENIGMA project is really piggybacking on a genomics consortium that we now have. And our genomics consortium coincidentally has 22 sites included, and there's genomic analyses are being done at 5 of those sites. So yeah, these are the sites around the world. So we have one in South America, a bunch in the US and in Europe, and so these are all sites. So this is all pooling of retrospectively collected data, and so the goal of this analysis is really looking at diagnosis of psychosis, as well as pediatric phenotype of psychosis proneness. And so for the genomic analysis for the overall 22q11 consortium, and this project was funded about almost 2 years ago. And so I won't go into a lot of detail about our genomic strategy, but it's pretty distributed across sites. A lot of the work is being done at Emory, and so they're doing whole genome. Steve Warren is heading that up and doing whole genome sequencing. And then a site at Leuven is really focusing on sequencing of the intact 22q alleles because it's a hemizygous deletion. And there's at Einstein, they're doing GWAS, and then so essentially looking at both rare and common single nucleotide variants as well as CNVs outside of the deletion region that may modify the phenotype. And so what we started was a few months ago when we started the project was really just collecting information across sites about their scan protocol and how many subjects they have available, and what psychiatric phenotypes these subjects have. And so we have 5 sites in the United States that are contributing data, 2 in the Netherlands and the Institute of Psychiatry in London. And then we actually, this is a little bit out of date because we now also have Australia on board, as well as, as well as Ireland and probably Switzerland as well. So so we are hoping to have, we have about half the data right now, and one of the nice things is that we're getting the raw DICOMs from all the sites, and so we'll be able to, we're processing it all at UCLA with the same protocol. So that'll be an advantage, I think. And so a lot of the sites do have similar to have a 3T Siemens scanner. Most of the sites also have DTI as well as resting state functional MRI, so that's something that we'll also be working on. And so this is just our abstract we just submitted to SFN based on, this is done by Frank Sun, who worked with us for a number of years and is now working at Loney. And so essentially it looks like using machine learning really has a lot of promise for classification of patients versus controls. One of the things that we found is that the data for 22q11 patients in terms of the overall pattern of volumetric deficits, cortical thickness, surface area, abnormalities relative to controls is very, very consistent across sites, and that's even looking at longitudinal data. One other thing I should mention is a lot of the sites do have longitudinal data, which will be nice to pool across sites. So I think, and this is what we found here, just based on using this machine learning classification based on our own site and then applying it to data from the Institute of Psychiatry data, we got a cross-site classification accuracy of 97%. So it's really encouraging, I think, for future analyses where we're able to include data from more sites. And so I think that's all I have. I realized that I was in such a hurry to put this together, I didn't even include what our major research questions are, but I think given the, one advantage of this data set is the genetic homogeneity, and so, of course, given the high rate of psychosis, we're really interested in some of the cross-disorder analyses, and particularly trying to identify patterns of neuroanatomic variation that relate to psychosis risk. And so looking at our data in comparison to Theo and Jessica's data will be great. And then, in terms of low-hanging fruit, one of the obvious things is really looking at deletion size in relationship to the degree of neuroanatomic variability, because that's something that we've tried to do in our own data, and because the atypical deletions are rarer, we really don't have the power to do it at a single site. So I think that'll be probably one of the first kind of genomic analyses that we will do. And so I'm going to stop there for questions. Questions or comments? Yeah.